Welcome to this virtual class on development of the spinal cord and also looking at spinal dysraphic disorders. This is part of a big lecture series on development and congenital malformations of the central nervous system. I usually give that lecture in 4 parts. The lecture on development and congenital anomalies of the central nervous system, I give it in 4 parts. But in this particular lecture, we're just looking at the second part of it, which is focusing on spinal cord and spinal dysraphic disorders. Maybe just to lighten you on the other 4, or rather the other 3 parts of the lecture series. In the first part, we looked at neurulation and neurotube disorders. So we talked about that at length. We saw what neurulation is, and we also looked at what are the neurotube disorders. And perhaps it is important that you have attended such a lecture on neurulation and neurotube disorders for you to be able to understand the development of the spinal cord. And so if you've not really read on neurulation or sat in a lecture of neurulation, I would suggest that first you do that first so that you're able to understand this second part of the lecture series that you're going to look at in this particular lecture, which is development of the spinal cord and the associated dysraphic disorders. In the third part of the lecture series on CNS development, I look at development of the brain and the ventricular system in general. And in the last part, I look at congenital anomalies of the brain. So here we're only focusing on the second part. Our specific objectives for this particular lecture would be, first, we will review the prenatal developmental phases of the central nervous system. I'm calling it a review because we mentioned it in the first lecture of neurulation. We'll also just have a brief review on what is neurulation, and we talk about primary and secondary neurulation. We'll also look at a brief review of the basic parts of the neurotube, as well as the cross-sectional anatomy of the neurotube. These ones have been captured well in the first lecture on neurulation and neurotube defects. So here, we'll just have a slight overview to help us understand what we want to do in this particular lecture. So in this particular lecture, in particular, we'll explain the derivatives and the differentiation of the cross-sectional neurotube at the level that is supposed to give us the spinal cord. And then we'll also explain why the spinal cord terminates at a higher level compared to the vertebral column. And we'll finish with looking at some of the developmental anomalies of the spinal cord, and that is where the spinal dysraphic disorders will also fall. So without wasting time, we can look at the first agenda, which is to review the concepts of prenatal developmental phases of the central nervous system. The central nervous system is described to develop in 4 phases. Some of these phases may overlap, but the events that take place are very characteristic and unique, and that is what helps us to outline the phases. The first phase of CNS development is a phase of dorsal induction, where we have formation of the neurotube. So basically, dorsal induction phase involves the process of neurulation. And at the end of neurulation, we have what we call a neurotube, which is the primordium of the central nervous system. After neurulation has taken place, Then you have ventral induction. Ventral induction involves molecular signal that make the rostral part of the neural tube to develop into the different parts of the brain. So that's what we call ventral induction. After that has happened, we have the third phase, which we call neural proliferation and migration. This is the time that now neurons undergo cells undergo mitosis to give us several cells, which are neurons and neuroblasts, and these neurons migrate. It's a period of neurogenesis when nerve cells are formed, and they don't just stay where they've been formed, they migrate to other locations. And so it is characterized by proliferation of cells and migration of cells from the wall of the neural tube. The last phase is a phase of myelination, which basically entails formation of the myelin sheath around axons of neurons. So I've said there are 4 phases. They may overlap, some of them, but at least there are distinct morphological events that take place in each phase. And that's why we outline into those 4. Now let's narrow down to the phase of dorsal induction, where we have formation of the neural tube. We call that process neurulation. Neurulation is a process of formation of the neural tube. And as I've told you, neural tube is the embryonic primordium of the central nervous system, which means the brain as well as the spinal cord. There are 2 types of neurulation. We have primary neurulation and secondary neurulation. And we are going to just have a brief discussion regarding the difference between the 2. The primary neurulation event itself takes place during the 3rd to the 4th week of human embryonic development, largely beginning from day 17 after conception, and is completed by perhaps day 28 after conception. That particular event, primary neurulation, requires the presence of the notochord. So the notochord induces the process of primary neurulation. Now, for us to understand what is primary neurulation and what secondary neurulation, we need to familiarize this image here. Let's take this to be the embryo. This is the cranial part of the embryo, and this is the caudal part of the embryo. From the caudal part of the embryo, we know we have what we call the primitive streak. The primitive streak forms the caudal midline. And at the end of the primitive streak here, we have a primitive node and a primitive pit. From the primitive pit, that's where the notochord usually forms, and it goes cranially. And so the message here is that the notochord does not span the whole of the embryo. It is only present on the cranial part. And that is why in cross-section, if you take a cross-section, the cranial part, then you see a notochord. But if you take a cross-section of the caudal part, we don't see a notochord. The neurotube itself spans almost the whole length of the baby. That means what then? If neurotube is going to be formed along the whole length of the embryo, the caudal end of the baby is unlikely to rely on the presence of the notochord for formation of the neurotube, because the caudal end lacks the notochord. That is why we have 2 types of neurulation. Primary neurulation refers to formation of the neurotube on the cranial end of the baby, and that one relies on the presence of the notochord. So we call it neurulation by notochordal induction. Secondary neurulation refers to formation of the neural tube in the caudal end of the baby. It does not rely on notochord, but it relies on mesenchymal condensation. And so we call it neurulation by mesenchymal condensation. In particular, the neural tube that arises from secondary neurulation will contain cells which are of mesenchymal origin initially, or mesodermal origin initially, while the neural tube that is formed from primary neurulation contains cells which were originally ectodermal origin. So in this image that summarizes primary neurulation, this is what happens. So if you look at this trilaminar embryo with the notochord in the middle and there, the notochord induces the ectoderm overlying it to become neuroectoderm. The ectoderm away from the notochord, therefore, is called surface ectoderm. The neuroectoderm itself undergoes thickening either by proliferation and increase in size of each cell. And that thickened neuroectoderm is what we call the neuroplate. So when we have the neuroplate undergoes folding like this. Perhaps that one is better. The depression within the neuroplate is called the neural groove, and the edges of the neuroplate, the elevated edges, are called the neural folds. Once the groove and the folds are formed, the two folds come together, fusing, usually from the level of the cervical region, then going in either direction, cranially and caudally in a zipper manner. And that will lead, therefore, to formation of something that looks like that, which we call the neural tube. The parts of the ectoderm that were not part of the neuroectoderm is surface ectoderm, and that one will give us epidermis of the skin. The parts of the neuroectoderm that are incorporated into the tube have formed the neural tube. However, there's some parts of neuroectoderm that don't become incorporated into the tube. And perhaps this image shows us that better. So if this is the neural plate that is now folding on itself, so that we have neural groove and neural folds, and that the folds come and fuse together. There's some cells of the neuroectoderm that are not incorporated into the tube, but they bud off like that and like that. We call these ones neurocrest. Neurocrest cells participate in formation of the peripheral nervous system, among many other derivatives that in the lecture on neurulation you may have captured. Neurotube participate in formation of the central nervous system, which is brain and spinal cord. So this is how the primary neurotube would look like. This is at the beginning of fusion process around the neck region. So the neurofolds fuse in a cranial and a caudal manner. Before the ends are fused, we call those open ends the neuropores. The cranial neuropore usually closes a bit earlier, while the caudal neuropore closes about 2 to 3 days later. By the time the caudal neuropore closes, we've now gone the end of the 4th week of human embryonic development. That is primary neurulation. How about secondary neurulation? Secondary neurulation does not require notochord, as we've mentioned. It requires mesenchymal condensation. It doesn't require notochord because, okay, let me put it this way. It has to use another mechanism of neurulation because the notochord is not present in the caudal end of the neural tube, in the caudal end of the embryo, yet you still need a neural tube in that region. So this neural tube will not form from a neural plate. It will form from mesenchymal condensation. Look at this image here, help us to capture that. I'd already shown it to you. So we want to see how the neural tube in this region forms now that we don't have an notochord. Let's take this to be a cross-section in that area. So that's ectoderm, mesoderm, and endoderm. So the first thing that happens is that the mesodermal cells here undergo condensation. And in the lecture on neurulation, we call that process mesenchymal epithelial transition. So the cells undergo condensation so that they form a solid linear structure. Imagine, you see this is cross-section, but if that is happening the whole length on the caudal region, so that becomes a solid linear structure, which you call the neural rod. The neural rod then undergoes cavitation through the process of apoptosis. And so what we eventually have is a structure with a lumen, which we call the neural tube. In this case, the secondary neural tube. That's how the secondary neural tube is formed, from mesenchymal condensation. If you look at these images, this shows you the region of primary neural tube, and this is the region of secondary neural tube, still undergoing neurulation. And perhaps this one has already gone neurulation, secondary neurulation, that's primary neurulation. Initially, the two tubes are not fused. But as time goes by, they become fused. So that usually by the end of the 7th or 8th week there, the secondary neural tube and the primary neural tube have now fused. The process of fusion of the primary and the secondary neural tube is sometimes given a name. We call that process junctional neurulation. The process of fusion of the primary and the secondary neural tube is called junctional neurulation. So that whole neural tube, the final neural tube is a neural tube that has been formed from both primary neurulation and secondary neurulation. And that neural tube is the one that will give us the central nervous system. The junction between the primary neural tube and the secondary neural tube largely correspond to the S1, S2 segments of the spinal cord, which means that even though we are making so much noise about secondary neurulation, it is only responsible for formation of the most distal small parts of the spinal cord. The rest of the central nervous system largely come from primary neurulation. That's why even the disorders we're going to look at are largely revolving around disorders of primary neurulation as opposed to disorders of secondary neurulation. So that is the process of neurulation. Now let's look at the basic parts of the neural tube and also the cross-sectional layout of the neural tube. I've already shown you this image showing us the primary neural tube basically with neurulation taking place, closure. And this one shows the cranial neuropore and the caudal neuropore just before they close. After they've closed, the cranial neuropore down here has closed, then joined with the secondary neural tube. And so we have one long neural tube. That long neural tube will now give us the different parts of the central nervous system. The cranial 2/3 of the neural tube give rise to the brain, while the caudal 1/3 of the neural tube gives rise to the spinal cord. Now, don't make that confusion of starting to think that because the spinal cord is very long, longer than the brain indeed, it should take a longer portion of the tube. That's not true. The truth is that we are looking at the mass of tissues being formed. Even though brain is short, definitely it has a bigger mass and even weight compared to the spinal cord. So the cranial 2/3 of the embryonic neural tube give rise to the brain, initially by forming 3 swellings, which we call the primary brain vesicles. It became induced to form those 3 swellings. And actually that is part of what we now call ventral induction, which we'll talk more about in the 3rd part of the lecture when you look at development of the brain itself. attention is taken towards this other end, then in this lecture, we'll be focusing on the caudal end of the neurotube. The caudal neurotube, the distal 1/3 of the neurotube, is the one that gives rise to the spinal cord, and that's what we'll be focusing on in this particular lecture. But in general, if you take a cross-section of the neurotube, the neuroepithelium is like this, somewhat stratified or pseudostratified columnar epithelium. These are neurocrest cells. So initially, that's what we have. Then as time goes by, we see some differentiation on the wall of the neurotube, as you can see in this particular image. I want to use the next image to show us better how this happens. The cross-sectional neurotube is characterized by rapid cellular proliferation, which makes the wall of the neurotube to thicken. And as the wall of the neurotube thickens, the lumen of the tube becomes narrow. As time goes by, the wall of the neurotube differentiates into 3 histological zones. The innermost zone is called the ventricular zone. The ventricular zone of the neurotube contains a group of cells which give rise to the other cells. So it's a stem cell population. Some people call the germinal zone, or the ependymal zone, or the ventricular zone. It contains a population of cells which are actively mitotic, giving rise to the other cell types. 2 groups of cells are perhaps present at this point. The lineage that give rise to the neurons are called neuroblasts, and the lineage that give rise to the supporting cells are called glioblasts. They're all present in that zone. After development has proceeded, and so all the cells have migrated to where they're supposed to migrate, remember the phase of neuronal migration, or rather neuronal proliferation and migration. After the cells have migrated, some cells will remain here. And the cells that will remain in that lining will become the ependymal lining of the ventricular system. So, or the central canal, the spinal cord, if you're talking about the spinal cord itself. In the brain, ependymal lining of the brain. Right. The second zone is the mantle zone. You can call it intermediate zone. This zone contains cell bodies of neurons and neuroblasts. This zone will give rise to the gray matter of the central nervous system, either spinal cord or brainstem, cerebrum, whichever. It will give rise to the gray matter, basically. And then we have the marginal zone. The marginal zone gives rise to the white matter. It contains axons of neurons, so giving rise to white matter. I want you to remember those 3 histological zones, because we'll be revolving around the march when you talk about differentiation of the spinal cord. Let's say that in a different way. 3 zones from inside: ependymal layer, which is also called ventricular layer or germinal layer, the layer of stem cells, but the layer that eventually become the ependymal lining. Mantle layer or intermediate zone contain gray matter. It's the layer that contains cell bodies of neurons. And then marginal zone contain axons of neurons, so it will call the future white matter of the central nervous system. Okay, with that in mind, I want us to narrow down to the mantle zone. As time goes by, the mantle zone become bigger and bigger. as we can compare in these two images. Other than that, the mantle zone also segment into some paired thickenings. We have a ventral paired thickening and a dorsal paired thickening. The ventral paired thickenings are known as the basal plates. The dorsal paired thickenings are known as the alar plates. So the mantle layer divides into the basal plate, the ventral thickenings, and alar plate, the dorsal thickening. Maybe this other image will be better in illustrating that concept. So look at that segmentation of the cord, or rather segmentation of the neural tube. This actually happens whether you are at the level of the spinal cord or you are at the level of the brain stem. Is the same concept. That the mantle layer segments into two thickenings. The ventral pair of thickenings called the basal plate. This basal plate represents the motor column of the central nervous system. So the cell bodies of neurons in this region will have motor functions. The alar plate, on the other hand, represent population of cell bodies of neurons that are sensory in function. So they call the alar plate the sensory column. There's a junction between the alar plate and the basal plate. That junction between the alar plate and the basal plate is called the sulcus limitans. So sulcus limitans basically represent the division between the motor column and the sensory column of the brainstem and spinal cord. The junction between the right and the left roof alar plates is known as the roof plate, and the junction between the right and the left basal plates is known as the floor plate. Having said that, we can now look at how that wall of the cross-section neurotube differentiates into the different elements of the spinal cord. So remember, we are looking at the differentiation of the distal neurotube. Distal third of the neurotube is the one that gives rise to the spinal cord. Let's start with the ventricular layer. So after the ventricular layer has served its functions of giving rise to cells that migrate away from it, the cells that remain will become the ependymal lining of the central canal of the spinal cord. The cavity of the caudoneurotube itself will become smaller and smaller. And so that cavity, we see it here, becomes the central canal of the spinal cord. And so the layer of cells that surround the central canal of the spinal cord is the ependymal lining of the central canal of the spinal cord. If this was at the level of the brain, then that cavity will become a ventricle, and that one will still be a ependymal lining of the ventricle. But we are at the level of the spinal cord, so that cavity becomes the central canal of the spinal cord, and the cells there become the ependymal lining of the central canal of the spinal cord. Let's keep the mantle layer a bit and talk about the marginal zone. The marginal zone contains axons of neurons. So because they contain axons of neurons, the marginal zone becomes the white matter of the spinal cord, And that's why the spinal cord will have its white matter outside and the gray matter inside. Remember, the converse is true in the cerebrum and cerebellum. And perhaps when you look at the development of the brain, we will see why in the cerebrum, the gray matter is outside to form the cortex. Now, coming back to the mantle zone. Mantle zone, we've agreed, has paired cubes or paired swellings. The ventral paired swellings, the basal plate, represent the motor column. So what does it become? It becomes the ventral horn of the spinal cord gray matter. Remember, the ventral horn of the spinal cord gray matter represents the somatic motor column. So what the basal plate does is to give rise to axons of neurons, give rise to neurons whose axons leave the spinal cord to innervate skeletal musculature. So note that the motor neurons arise from inside here. Their cell bodies are inside here. The axons grow out to follow skeletal musculature. Apart from giving rise to somatic motor neurons, which are housed within the ventral gray matter or the ventral horn, the basal plate also gives rise to the intermediate horn of the spinal cord, or what we call the lateral horn. Remember, lateral horn contains cell bodies of preganglionic autonomic neurons. So those ones go to target organs of the autonomic nervous system. In simple terms, the intermediate horn has a visceral motor function. So whether somatic motor or visceral motor or autonomic motor, we appreciate, therefore, that the basal plate has a motor function, so to give rise to efferent neurons which leave the spinal cord. They could be going to skeletal muscles, so we call them somatic motor, or they could be going to smooth muscles, cardiac muscles, and glands. We call them visceral motor. Of importance, visceral motor is lateral horn, and somatic motor is ventral horn. The alar plate, on the other hand, represents sensory column. This is the one that receives sensory nerves entering it. And so in the spinal cord, the alar plate becomes the dorsal gray horn of the spinal cord. So what I've said in the previous slide is also captured in this particular image. What I want to repeat here is that for the dorsal horn, remember it's sensory, so it will be the nerves entering. The dorsal horn does not give you sensory nerves that leave, no. It will be nerves entering. So remember, the nerves that enter are of neurocrest origin from our previous lecture on neurulation. Those cells are of neurocrest origin, then they invade the spinal cord from the posterior horn. But for basal plate, the neurons arise from the basal plate outwards. And so a spinal nerve is a nerve that will contain both sensory as well as motor functions. It's a mixed nerve. This image captures that concept of neurocrest cells, giving you the neurons that enter through the sensory column, so basically sensory nerves, while the basal plate give you neurons that leave from the front ventral horn for somatic motor, or from the lateral aspect, lateral horn for visceral motor. And that a spinal nerve is a mixed nerve either way. Now let's look at why the spinal cord and the vertebral column are not of the same length. Let's start by looking at this. Initially, the spinal cord and the vertebral column are of the same length, actually, that story stands up to around the 3rd month of pregnancy. Now, from there, the rate of growth of the spinal cord is different from the rate of growth of the spine itself, the vertebral column. So what happens? The vertebral column will outgrow the spinal cord as time goes by. Remember, the spinal cord is attached to the brain. And so if the spinal cord is not increasing in length as fast as the vertebral column, it will continue to hang upwards as the person grows, because it is fixed from the cranial aspect because it's attached to the brain. If there was something that was to forcefully attach the spinal cord down so that it can't really hang in the manner it does, that can give us some spectrum of malformations of the brain, which are due to traction of the spinal cord. And so we may have some intracranial manifestations of those spinal cord lesions. And in the lecture on neurulation, we call those ones Chiari malformations. So here, let's start again. If the spinal cord is the full length of the vertebral column at the beginning, and remember, spinal cord gives you spinal nerves, so you'll have spinal nerves exiting corresponding with their vertebral levels ideally. But as the spinal cord shortens or vertebral column elongates faster, usually around the 6th month of intrauterine life, the spinal cord is now reaching the level of S1 vertebra. That means it has hung a bit. And for that reason, the spinal nerve roots that were originally almost horizontal like that will now be pulled upwards in this manner, so that they'll be having a vertical orientation first before they have a lateral orientation or a transverse orientation. At the time of birth, what's the situation? When a baby is being born, the spinal cord reaches around the level of L3 vertebra. Again, look at our nerves. They're then elongated more, so coming down in an oriented manner, then transversely. How about in an adult? In an adult, the spinal cord ends at the level of L1, L2 junction most of the time. And so the understanding of the nerve roots still stands at that point. Now, the imagination I've given you is that of just a single nerve root. Now imagine all the nerve roots below that region, L1, L2, L3, all the way to coxigeo 1. Imagine all of them, and they're all paired. How will that appear? It will appear like a group of neurons that are descending in the lower part of the cord first before they exit. And maybe this one captures that better. This group of spinal nerves at the lower end of the spinal cord constitute what we call the cauda equina. It means the tail of a horse. And it actually looks like the tail of a horse. So those are the reasons why the spinal cord is shorter or has a proximal termination. Or we can look at it this way. Those are the outcomes of the change or difference in growth rate between the vertebral column and the spinal cord. Because the vertebral column grows faster than the spinal cord terminates proximally. And because of that, there is cauda equina. We have a cauda equina. Okay. One last part, therefore, is to look at the development and congenital malformation of the spinal cord. Some of these were also explained under neurotube defects, and so I'll not spend much time on them. We use the term spinal dysraphism to describe such lesions. Remember, we said that these lesions can affect many things. At the level of the spinal cord, they affect the vertebral column. They affect the spinal cord itself. They may affect the skin and surrounding connective tissue. Generally, those are neurotube defects. Now, the ones we call spinal dysraphism are specifically neurotube defects that occur at the level of the spine and spinal cord. This is to distinguish them from the cranial defects. So what are examples of these neurotube defects? We have spina bifida disorders, where the vertebral column has not closed, or let me say, the neural arc has a defect. Remember, this is the vertebral body. This is a pedicle. We expect lamina and lamina to join that way, and then a spinous process. Here we are lacking the lamina and we are lacking the spinous process. It appears like there are two projections from this vertebra, and there's a defect there. And that's why we call it bifida, spina bifida. In this first one, the swelling is not there. And that's why we call it spina bifida occulta. In this two and third one, there are some swellings. And depending on the content of the swelling, we can call it meningocele or meningomyelocele. Maybe this one shows better the concept of spina bifida disorder, that one. Where there is no swelling, we call it spina bifida occulta, from the word occult means hidden. Then these are the ones were originally called cystica, but that term is not very helpful. So you'd rather call them some specific terms like meningocele for this one. It means that there's a defect, then the CSF and the meninges have popped through that defect, giving us a swelling. That's what we call meningocele. In this other one, in addition to the meninges and CSF, some neurotissue is also present. It doesn't have to be the whole cord as this one is showing. As long as some neurotube is present, we call that myelomeningocele or meningomyelocele. So this shows you spina bifida occulta, where we're just seeing a dimple and some hair growth and hyperpigmentation. This one shows you either meningocele or myelomeningocele, depending on the content of that swelling. You may have situations where the neurotube did not close completely, and so you have an open neuroplate. That's what we call rhaschisis. If it is involving the whole of the neural axis, which means that both the cranium as well as the spinal cord is affected, then we call that craniorachisis. The term rachi means spine. Schisis means split or open. So craniorachisis means that the skull is affected, the vertebral column is affected. How? They're open. Other forms of spinal dysraphism may just involve splitting of the spinal cord into two, like what we see here. And that is what we call diastematomyelia, split spinal cord. Other malformations that can be more common as well include this one. So this is an MRI image of the cervical spine so that we are able to see the spinal cord. Then we see this dilated thing here, which has the same signal as the CSF within the fourth ventricle up there, or CSF within the subarachnoid spaces here and there. It means that that swelling is therefore most likely fluid. So that fluid within the central canal, you may have fluid within the central canal, or you may have fluid parallel to the central canal. We use a common term, hydrosyringomelia, to refer to any of those two entities because it is difficult to distinguish when you have a fluid pocket within the spinal cord. Is it inside the central canal? That's what we, or is it parallel to the central canal? So because it's difficult to distinguish the two, we just have a common diagnosis of syringomelia. I want you to think through that and ask yourself, so which one will we call hydromelia? And which one will we actually call syringomelia? Out of the two entities, think about it. This other image shows something unique. I want you to follow it up. Look at that. This dark thing represents the spinal cord, all the way to somewhere there, where it terminates. But which vertebra is this? This is the fifth or the last lumbar vertebra, and that's the first sacral vertebra, or the second sacral vertebra. So what do you notice about our cord? The cord is not terminating at L1, L2 junction there, where it's supposed to terminate, but otherwise has extended all the way down to the sacral spine. This cord is not really shortening. Why would it not shorten if something is holding it down? So such a cord is what we call tethered cord. Tethered cord is a cord that has been held down so that it can't shorten. What's the problem with this? If there's tethered cord, you may now be pulling the brain through foramen magnum. And that can give you other anomalies, but basically anomalies of brain herniation. Great, so that is spinal cord development. The third part of lecture on congenital development and congenital malformations of the central nervous system will focus on development of the brain as well as that of the ventricular system. So for now, we'll stop there. Thank you very much.